Well, hello everyone, and welcome back to Human Biology here at Chaminade University. Today we will be discussing the digestive system. So this is just an overview of the digestive system. We're going to start with the mouth, which is where we're going to take in food. We're going to start the process of what we call mastication or chewing here in the mouth. This is going to be our first set of mechanical and also chemical digestion. So you can see we have here the salivary gland, which is going to introduce saliva, which has an enzyme called amylase. Amylase breaks down amylose and therefore is going to be an enzyme. Anyway, in the mouth here we're going to chew and then we're going to swallow this bolus of food. And as it goes down through the throats, we're going to enter into the pharynx. The pharynx is a funnel-shaped organ here at the back of the mouth, which then enters into the throat. The throat has both the esophagus and the trachea, but obviously food is going to go down the esophagus here. Swallowing down the esophagus all the way until we get down into the stomach. Here's the stomach. We're going to have multiple different layers of muscle tissue here, actually an additional layer of muscle tissue that's not seen anywhere else in the digestive tract, which allows us to cross-sectionally digest this. We also are going to have, so we not only the muscle, which is going to be mechanical digestion, but chemical digestion starting here too. So we're going to be secreting a bunch of different enzymes that are going to start breaking down. We have lipase that breaks down lipids. We have the special things that are going to break down proteins, as well as hydrochloric acid, which is going to break down whatever it touches, right? So that's going to be secreted in the gastric juices here. That means that here we're going to have a pH of about 4. And as we enter into the intestines, we're going to have to bring that pH up. So here we are entering into the intestines. We're going to want a pH of about 7. So what that means is that although you can eat an entire cheeseburger, an entire meal in one sitting, and it'll all fill up in the stomach is only going to dole out about a teaspoon or 2 of food at a time. That means that the intestines will have the chance to modify the pH of that food and bring it up to about 7 so that we can continue the process of chemical digestion in the intestines. So when we first enter the intestines, we'll enter into the duodenum. That's here. Then we have the jejunum here. Then the ileum. The ileum is going to lead into the cecum, here through the Ileocecal Canal. The cecum is going to be the beginning of the large intestines. Here you see this little dangly organ. This is called your appendix. It's a vestigial organ that we no longer utilize, although it does tend to become inflamed and cause us to have surgeries to have it removed. Then we head up through the cecum. We're going to hit the ascending colon, the transverse colon, the descending colon, and then we're going to end up with the sigmoid colon, so named because it looks like a little sigmoid. Then we get the rectum, which feces is going to be stored until it is going to be able to be expelled out through the anus. So that's going to be an overview of the digestive system. Now, the digestive system has multiple different sets of organs. We have direct digestive organs and accessory organs. So some of the organs are going to directly come in contact with food, and some things like the pancreas, for example, are just going to be accessory organs, which are involved in chemical digestion or secretion of things into the digestive tract, but never come in contact with the food itself. The process of ingestion is what we're going to start with. Again, ingestion is when we're taking in the foods, the process of taking the bite of that cheeseburger. Then we're going to masticate. What does that mean? That means to chew. So the process of mastication is the first start of mechanical digestion, when we're going to start physically mixing and breaking down the food into smaller pieces. But again, throughout the digestive tract, we have both mechanical and chemical digestion. So chemical digestion is going to be when we have enzymes that are involved in breaking down the biomolecules into smaller and smaller biomolecules that can then get absorbed across the intestinal lining. So the first part of the digestive system is going to be secreting a lot of things into that food. So that bolus of food that you start off with as one bite is going to double or maybe triple in size by the time it gets released out of the stomach. Why? Because we're going to have a ton of things secreted into it. Water, ions, mainly salts, gastric acids and gastric juices, enzymes, bile and bicarbonate ions. All of these things are going to be coming into this food in various regions of the gastrointestinal tract. So gastric acid, that's stomach, bile and bicarbonate, that's early intestines. But the point is, all of these are going to be involved in aiding digestion. So we're secreting a bunch of liquid in, and then we're going to have to pull that liquid back out. So that absorption is going to occur in the intestines. Mainly the absorption of nutrients is going to occur in the small intestines, and the absorption of water is going to occur in the large intestines. But absorption is basically going to be the process of moving the nutrients and the water and the ions from the lumen of the digestive tract, remember that's the inside of the hose that's traveling through us, into the bloodstream, where it's going to then get delivered to the different parts of the body. At the end of the day, we're going to end up with matter that we can't use. Undigested matter, that's called feces, and that's going to be excreted again through the anus through the process of defecation. Okay. So this is just kind of an overview of the digestive system. First things first, food's going in. You're going to take a bite, right? Food and water go in through the mouth, and the process of mechanical and chemical digestion occurs. Again, mechanical or mastication, chewing. Chemical is going to be the release of like amylase, which breaks down amylose, which is going to be the very first set of chemical digestion that occurs. Then we swallow. We swallow through a cool little tube called the esophagus that has muscles that constrict in repeated order. So 1 to 2 to 3 to 4 to 5. What that means is that you can swallow even upside down because the next muscle in the row takes the food and swallows it down, forcing it into the stomach. There's a nice sphincter right here, the lower esophageal sphincter, that's going to allow the food into the stomach. And when we're in the stomach, we're going to have multiple different processes occur. So as you can see down here in the key, we're going to see whether or not we have ingestion, mechanical digestion, chemical digestion. Again, in the mouth, we're going to have ingestion, mechanical digestion, chemical digestion, and secretion, right? We're going to have secretion of different things coming into our saliva. In the stomach, we're going to continue on. Again, ingestion is over, but we're still going to have mechanical digestion, chemical digestion, secretion, and absorption occurring inside the stomach. From this point on, we're going to have a really low pH, so we're going to have to moderate how we secrete it into the small intestines because the small intestines is going to want a normal pH of about 7. So although, again, we're able to eat a whole meal at once, only a small amount is going to be doled out into the small intestines at a time. As we're traveling through our small intestines, we're going to have things added to the small intestines from the liver, the gallbladder, and the pancreas. These are all accessory organs. The liver creates bile. The bile is stored in the gallbladder when it needs it. The gallbladder can squeeze and secrete it into the small intestines. The pancreas is also going to make pancreatic enzymes, etc., and bicarbonate that are going to be secreted in the small intestines, all to help bring that pH back up to 7, as well as to help continue the process of chemical digestion. So we're going to have mechanical digestion, chemical digestion, secretion, and absorption occurring here in the small intestines. And in the large intestines, we're focusing mainly on absorption, specifically the absorption of water, right? By the time we've gotten to the point where we've eliminated everything out of that biomatter and we're ready for the feces to be defecated, that's going to, again, end up happening out through the rectum through the process of defecation, right? So that's an overview of the digestive system, and we're going to break it down as we travel through here. So the function of the digestive system is to physically and chemically break down food you have ingested, to absorb all the digestive nutrients into the bloodstream, to remove undigested waste, and to produce all sorts of different biochemicals, including intrinsic factors, that are required for the absorption of biomolecules. The intrinsic factor specifically is required for the absorption of vitamin B12, but there's a lot of different things that we are going to be creating that are required for the absorption of particular biomolecules. We also are going to be housing symbiotic bacteria that are good for us, the normal flora that are found in the large intestines that help us to create different biomolecules and nutrients. If we're looking at the tissues of the digestive system, we have many different layers that we're going to see. If we're looking at them throughout, starting with the throat, working our way through the stomach, and then through the intestines, we're going to see epithelial layers, lamina propria layers, muscle layers, etc. These are going to change a little bit as we travel through, and we'll talk about how, but they're all going to serve the same roles and still be present, just maybe in different strengths or weaknesses depending on which region of the tract we're in. The epithelial layer is going to function in absorption. The lamina propria is going to be our vasculature, our nervous system, and our lymphatic tissue. We haven't talked about lymphatic tissue yet, but lymphatic tissue is going to serve to pick up extra fluid that didn't quite make it back into the blood supply and run it past what we call lymph nodes to help to be able to digest any sort of foreign invaders. But for the digestive system, it's also going to serve to help pick up the fats that are going to be absorbed across the intestinal lining to be able to drop them specifically back into the bloodstream through a whole separate route than things that are going to be water soluble. So it also has a special specialty here within the digestive system. The muscular layer is called the Muscularis mucosal layer. We have the smooth muscle layer. We also have mucosal plexus or the submucosal plexus, which are going to be nerves that are going to stimulate digestive enzymes and other secretions. And we have another muscle layer called the external, so the Muscularis external. It's going to be comprised of circular muscle, Myenteric Plexus is going to be controlling the circular muscle and the longitudinal muscle. So the circular muscle is going to help mix intestinal contents. The longitudinal muscle is going to move food throughout the GI tract. And we're going to get another layer of muscle that's only found in the stomach called the oblique layer that's going to pop up here. And again, the Myenteric Plexus is going to control those coordinations as well. So we have these plexus here that are shown here in the bold. That's going to be your nervous tissue that's going to be responsible for the control of digestive enzymes for the submucosal layer and the contractions of the muscle for the Myenteric Plexus. All right. We also have a serosa layer, which is a connective layer that's going to line the entire outside of the GI tract. So this is what we're going to see in terms of the layers of the GI tract as we travel through. We will see it change a little bit as we go through different layers. Again, the stomach will get an extra layer of muscle, for example, but we're going to see it generally remain in place as the same. So we're going to have two muscularis layers. Here's the longitudinal and the circular. In between them, we're going to have this myenteric plexus. Again, the myenteric plexus controls the innervation of these two. Inside that, we have the submucosal layer. The submucosal plexus is going to control secretions. So here's the glands and the glandular secretions. Underneath that, here we're going to end up with the muscle mucosa layer. It's going to have the lamina propria, the epithelial lining, and this is going to be lymphatic, glandular secretion, the submucosal glands. These guys here are the lymphatic tissue. So this is the overview of what you're going to see in terms of the layers of the digestive tract as we travel through the digestive lining. All right, so these are the major tissues of the digestive system and how they are held together. There's this region here called the omentum and the mesentery. The greater omentum and the mesentery are going to be connective structural tissues. This greater omentum is going to line the entire of your digestive tract, keeping your intestines and etc. held together. And if we pull that greater omentum upward, you're also going to be lifting that transverse colon, which normally runs like this. Now it's flipping upward with the omentum. That is going to allow us to visualize the mesentery here. This mesentery layer is going to be a connective layer that holds the transverse colon and the descending colon, etc. in place. We'll also see here the ileum has been pulled laterally, so moved over. All of that would normally be here, just so you're able to see that mesentery layer. So it's all going to be connective tissue, right? So the greater omentum is going to be adipose tissue, and the mesentery is going to be holding those abdominal organs in place. All right, so here's the application question for this chapter. So as we continue through the rest of this lecture, I want you to consider what happens to the food that gets eaten. This was eaten by a girl named Martha. She's eaten beef, potatoes, peas, carrots, onions, and celery in a broth. The question is, where in the digestive tract is mechanical secretion or mechanical digestion and or chemical digestion taking place? We've already kind of covered that, but think about that as we go forward. And then also, which foods contain the most of which of the biomolecules? Proteins, fats, starches, and fibers, for example. What enzymes break down each of those different biomolecules in her meal? And what hormones are we going to be producing when we have eaten enough such that we feel satiated or full and we don't want to have another bite? All right, so let's move forward. We're going to talk about the organs of the digestive system, starting with the mouth. The mouth has two major bones. The mandible is the jaw. The maxilla is going to be the upper layer where your teeth are going to be interacted, right, like your cheekbones and your teeth. The gums are going to be called gingiva. So when they get infected, it's called gingivitis. The tongue is a muscle. It's going to help move food around. It's going to also allow us to speak, right? So it's going to allow us to communicate with each other. It's also going to allow us to swallow and has taste receptors to allow us to taste multiple different tastes, including salt, sweet, bitter, sour, and what we call umami or savory. At the very ends of our mouth, we have the uvula. The uvula is going to block our nasal passages from being in direct contact with our mouth. It's that little dangly thing at the back of your throat. If you've ever seen Yosemite Sam scream, you can see it dangle, right? In the pharynx, which is in the actual back of the throat as you're starting to swallow, we have something called the epiglottis. So the epiglottis is normally open so the breathing can occur. That means that every breath you take, the air goes in and out of the trachea and into the lungs. However, when you swallow, that epiglottis flips forward to close off the trachea so that the food's going to go down the esophagus. We're going to be using muscle contractions throughout the entire GI tract, starting with the ability to swallow all the way out through the defecation reflex. It's going to be called peristalsis, basically pulling things forward from one part to the other. Okay, so we have multiple different sets of salivary glands. Salivary glands are going to be found in our parotid, submandibular, and sublingual. These are paired glands, so there are 6 of them. And saliva is mainly water, but it also has a lot of enzymes that are going to be bactericidal, such as lysozyme, which is going to kill bacteria. It also has enzymes that are going to start breaking down sugar, like amylose. And it's also going to be secreting mucus to help make everything nice and smooth and lubed up, particularly if you eat something tough like a cracker, so that it doesn't scratch up the inside of your throat as you swallow, right? So it helps moisten and lubricate that food. Also going to start digesting starch, amylase, and fats. So lipase is another fat that's found, salivary lipase. It's another enzyme that's found to help break down fat. We'll see gastric lipase as well. So lipase just means that it breaks down fat, but this one's going to be salivary lipase because it's found in the saliva. And it also is going to help break down and dissolve food. We're only going to be able to taste dissolved food molecules. So once things get broken down into small enough molecules to reach the taste buds to hit the chemical sensors, then we're going to be able to taste that food. All right, so this is the mouth. As we can see, we have multiple different teeth. So we're going to have the incisors here. They're going to be at the front. Then we have the canines, the little sharp ones that have the pointers. Then the premolars, so that we're going to be starting to get wider. And then the molars, which are the wider ones. Looking inside, we obviously are going to have the tongue. We have taste buds. At the very back here, we have the soft palate, which is separated from the back of the throat by the uvula here. And these guys here, these are gums or gingiva. All right, so as we swallow, right, we're going to swallow by putting the bolus of food at the back of our throat, putting the tongue upward, and then allowing the food to move down into the pharynx. The pharynx is going to be a funnel-shaped organ at the back of the throat. And again, this is the epiglottis, which is normally open so that air can go in through that trachea. But as we swallow, we're going to close that epiglottis. It's called the epiglottis because it's going to close off the glottis. So we're going to close off the glottis, and when the glottis is closed, then the bolus of food can travel right on down into the esophagus. As I mentioned, the esophagus is going to have a series of muscle contractions. So we have relax and then contract muscle so that you're able to swallow even upside down. And the contraction of these circular and longitudinal muscles is going to bring the food down past the lower esophageal sphincter into the stomach. All right, so here is the stomach. They call it the gastroesophageal sphincter. You may also hear of it as the lower esophageal sphincter, so the GES or the LES. They're both the same thing. And then this is going to feed into the stomach. The stomach is going to have a region that's going to be called the fundus, which is the area opposite the opening here. It's going to be the serosa layer. And then the region called the pylorus, which is going to be closest to what we call the pyloric sphincter, which makes sense. That's the connection between the stomach and the intestines. Keep in mind, this pH is 4, this pH is 7. So we can't have a flood coming out here. We will destroy the intestines. We also can't have this coming back up here. If we do, it's called gastric reflux syndrome, and it means that you're going to end up with heartburn. We're going to have things popping up into that throat that include our gastric juices, which again have a very low pH and are going to have a lot of hydrochloric acid in there. All right, so let's look at the muscle layer. So we have the longitudinal muscle and the circular muscle, which we've always had, but in this case, we also have an oblique muscle. So longitudinal running this way, circular running that way, and oblique running diagonal. And that allows us to have three-dimensional multiple different ways that we're able to contract the stomach to allow for what we call gastric churning, or the churning of the food inside this gastric chamber. Here we have the submucosal layer, which is going to have the mucosal layer on top of it. And these here are called rugae, and they allow for the stomach to expand. So the stomach can hold up to a gallon of food. More if you're a hot dog eating contest participant. Just joking. But really they eat an enormous amount of food because of the fact that these are elastic. So they're able to contract down, and then they open up when these rugae are able to stretch out when you have a lot of food in here. Again, we're only going to, although we might eat a gallon of food at once, we're only going to be secreting out about a tablespoon or two at a time out through this pyloric sphincter. That's all the duodenum is able to handle in terms of getting the pH back up into 7. From here, we're also going to interact with things that are like bile that are coming from the liver and the gallbladder, and also from bicarbonate and pancreatic enzymes coming from the pancreas. All right. Okay, so the bolus of food enters into the stomach, and then we have secretions that are going to come in. Now it's going to be called chyme, right, or chyme. So the stomach is divided into 3 major sections: the cardia, the fundus, and the pylora. All right, so here's the fundus, here's the cardia, the area that's just coming in. This is the body, and then this here is the pylorus, or the region that's going to feed out through the pyloric sphincter into the duodenum. Okay, the lower esophageal sphincter, we also just saw it called right here the gastroesophageal sphincter. Those are the same thing. It's going to prevent the stomach contents from going back up into the esophagus, right, while we are undergoing pretty intense mechanical digestion. What do I mean by that? I mean gastric churning. I mean that all 3 of these layers of the muscle are going to be contracting in different timing to be able to make sure that we have churning of all of the food inside there, right? And again, that pyloric sphincter is going to hold the chyme in the stomach until it's ready to enter into the small intestines. And we have those rugae that I just showed you that allow the expansion of the stomach so that you can eat a whole gallon of food all at once. Inside the stomach, we have specialty cells like goblet cells. They're so named because they look like goblets. They're going to produce mucus. The mucus is going to help protect the stomach lining. Remember, the stomach is going to be secreting things like hydrochloric acid. Hydrochloric acid is not good for cells. So the cells that are found inside the stomach lining need to have this mucus on top of them like a blanket to protect them from the acid that's going to be digesting the food inside of there. All right? We have specialty cells that are called chief cells. Chief cells are going to produce a, anytime you see inogen, you know it's going to be a precursor. So pepsinogen, ogen means precursor, it's a precursor to pepsin. So pepsinogen gets converted into pepsin, which is the activated version of the protein, when we have HCl present. As soon as HCl converts pepsinogen into pepsin, pepsin starts digesting proteins in the stomach. Pepsin works very well at this pH of 4. As soon as it enters into the intestines, where it's going to be replaced by trypsin, and pepsin is no longer going to work because the pH is going to jump to 7 and pepsin cannot work at a pH of 7. But where does all this hydrochloric acid come from? They come from a special type of cells called parietal cells. Parietal cells are going to produce hydrogen on one side and chlorine on the other to create hydrochloric acid. Now, it's not going to spew hydrochloric acid out of one fire hose, more like it's going to spew hydrogen out of one side and chlorine out of another directly at each other, such that they're created inside the stomach but not inside the cells. And the hydrochloric acid, again, that's going to be responsible for activating pepsinogen into pepsin, and also, because of the low pH, kills any pathogens that we may have introduced by eating something like a soft cheeses or undercooked meats, etc. Parietal cells also produce that intrinsic factor that we talked about that helps make vitamin B12 able to be absorbed inside the small intestines. All right, so I said it can eat a gallon. I was a little bit exaggerative. The stomach can hold about 2 liters, right? But it has an empty volume of about 75 milliliters. So it goes from about 75 mils to about 2,000 mils, which is what 2 liters is, when we can eat an entire meal. And that's quite a lot. It's about half a gallon, in fact. And when we contract the stomach muscles, we're going to be able to do that by way of the parasympathetic nervous system. Now, we haven't discussed the nervous system yet in entirety, but it's split up into sympathetic, which is fight or flight, and parasympathetic, which is rest and digest. So as you can imagine, since we are a digestive system, we're under the parasympathetic control of the nervous system here. We are also under the influence of nitric oxide and serotonin. These are going to help be responsible for the relaxation of stomach muscles. And the contraction of the stomach muscle is going to be initiated by what we call pacemaker cells. All right, now in order for us to be looking here at this area here, including these accessory organs, this liver has been kind of flipped up. So you can't, anyway, I'm digressing. These are the accessory organs of the digestive tract. As I mentioned previously, the accessory organs are going to be anything that does not come in direct contact with food, but just is going to secrete things like bile, for example. So here is the liver. The liver is going to create bile. The created bile is going to be stored in the gallbladder. Okay, the gallbladder is going to be able to release, so it can squeeze whenever, and secrete large amounts of bile directly out through the hepatopancreatic ducts here into the gallbladder. From the gallbladder into the intestines. Here's the duodenum of the intestines. So bile comes out of the gallbladder into the intestines here through the hepatopancreatic ducts. And it does so that we can increase the digestive capability of the kind that's coming out of the stomach. We're also going to have the pancreas as an accessory organ. It's nestled right on in here inside the duodenum. The pancreas is going to have a main pancreatic duct that's going to secrete out through the hepatopancreatic duct, as well as an accessory duct that secretes directly out into the small intestines itself. Pancreas is going to secrete pancreatic enzymes as well as bicarbonate. And bicarbonate, as you know, is a buffer, which is going to help bring our pH back up to the 7, which is what's necessary for digestion in the intestines. So again, accessory organs are going to include the liver. There's a falciform ligament of the liver, which is split up into 2 different regions in terms of ducts. Here we have the left hepatic duct and the right hepatic duct draining into the gallbladder. The gallbladder is connected by the cystic duct into the common bile duct to feed into the hepatopancreatic duct directly into the intestines. So again, that's going to include the liver, the gallbladder, and the pancreas. All right, so the liver is going to be the second largest organ. The largest organ in your body is your skin. The main cell types of the liver are going to be including the hepatocytes. Hepatocytes are going to directly absorb nutrients after digestion because of what's called the hepatic portal blood flow. So nutrients are going to come directly from the intestines into the liver. The liver is going to be responsible for sorting and storing the nutrients, and delivery of the nutrients will then be sent out through the vascular system. We also have Kuffer cells. Kuffer cells are immune cells inside the liver that are responsible for destroying pathogens and also breaking down any dead blood cells or other dead cells that are found. So Kuffer cells are resident immune cells found in the liver that are responsible for making sure that anything that comes in that hasn't already been killed by the HCl in the stomach, etc., is going to be destroyed before it gets sent out through the vasculature. The liver also is going to be responsible for creating bile. Bile is responsible for the digestion of fats, and it's going to be stored in the gallbladder. So it's synthesized by the liver in a slight little drip. You can picture like it drips out of the faucet, and there's a bucket under the faucet. That's the gallbladder that's going to connect it. Going to collect it, I'm sorry. Liver is going to be storing nutrients during its phase that's absorption. So absorptive phase is going to be stimulated by insulin. Remember, insulin is going to be responsible for regulating your blood sugar. If your blood sugar gets too high, insulin's turned on to take the blood sugar out away from the blood and store it in some places, including the liver. It's going to be responsible for converting glucose into glycogen in the liver. That's called glycogenesis. So genesis being creation, glycogenesis is the creation of glycogen. And we also are going to be converting glucose and amino acids into triglycerides. That's called lipogenesis, or the creation of lipids. And it's producing lipoproteins, which are responsible for transporting things like fats, for example, from the liver and storing them in adipose tissue. Liver also is going to store excess fat-soluble vitamins, which makes sense, right? We've got a lot of lipids we're creating here. We're also going to have a bunch of lipoproteins, so it's going to be a lipid-happy region. So we're going to store fat-soluble vitamins such as vitamins A, D, E, and K, which is why in olden times you would eat the liver of the animal, or we would make liverwurst because it has a ton of vitamins in it from whatever animal you've slaughtered. It's also going to be responsible for storing minerals and trace metals. Liver is going to have, again, two different regions, or two different phases. The first phase we just talked about was the absorptive phase. The second phase is the fasting phase. The fasting phase is stimulated by what's called glucagon, also by epinephrine, thyroid hormone, and growth hormone. All of this happens when there's not a bunch of sugar in the bloodstream. In this case, we want to be creating sugars. So we're going to take glycogen and make it into sugar by glycogenolysis, lysis being breaking down. Breaking down glycogen is glycogenolysis. We also can convert those triglycerides back into glucose. You'll note these are all the same things that we just did, but in reverse, right? We just converted glucose into glycogen. We just converted glucose into triglycerides. Now we're breaking them back down during the fasting phase to be able to put blood sugar back to normal, right? We want to put glucose back in our bloodstream because it's dropped too low. And if we get too low of our blood sugar, right, we can get sick. The liver also converts fatty acids into ketones, which can be converted into acetyl-CoA, which, if you know anything about the Krebs cycle, are going to be involved in the creation or production of ATP. And we're also going to be producing urea from the breakdown of protein products. So the liver, again, it does a lot of things. It's also responsible for breaking down red blood cells. The spleen is also responsible for this, but the liver is going to be the primary responsibility here. And a component of the red blood cells is called hemoglobin. Hemoglobin carries oxygen to the cells. Hemoglobin is going to have heme as a component, which is going to be ferrous, or has it's going to have iron in it. And that can be converted to bilirubin. Bilirubin can be added to the bile and excreted during the defecation. Okay, so anything that's extra that's from the red blood cells, extra heme, is going to be broken down into bilirubin and then excreted. The liver also breaks down organic and inorganic toxins, things like drugs, alcohol, foreign toxins, etc., and adds the breakdown process to bile for excretion. So while I said previously that you might slaughter the animal and eat its liver, you want to be really careful that you're not going to be eating the liver of an animal that has had a bunch of foreign toxins in it from its environment. So domesticated animals, yes, but probably not wild animals, right? The liver also is going to excrete excess trace metals that we may have been in contact with that we don't need, and synthesize plasma proteins, so proteins that are found in our blood supply, in our bloodstream, such as albumin. Albumin is really important for the regulation of our osmotic or polar osmolarity. It also is going to create binding proteins that are going to help us transport fat-soluble hormones in the bloodstream. You might remember that the bloodstream is not very friendly to fats. In fact, it's going to be what we call hydrophilic, which means it interacts well with water. Fat-soluble hormones are going to be hydrophobic, so we want to have a specific type of proteins. These are going to be amphipathic proteins that are going to help transport fat-soluble hormones through the bloodstream, which is not always necessarily going to be receptive to that by itself. The liver also synthesizes what's called fibrin fibrinogen. This is going to be responsible for blood clotting response, and angiotensinogen that is involved in regulation of blood pressure. It also is going to activate vitamin D, which is going to be a precursor produced via sun exposure in this layer of skin. And vitamin D is really important because it regulates the immune system as well as the regulation of calcium homeostasis, which is really important for your bones. And the liver produces IGFs, which are insulin-like growth factors. Growth factors are really important. They respond to things like growth hormones and allow our cells to decide when to divide and when not to divide. It also is going to secrete complement proteins that are involved in the immune response. So the liver does a lot of different things. But what we really know it for and know it for well is the creation of bile or bile salts. So bile is required for the digestion of fat. So here's the bile salts. And bile has both a nonpolar side and a polar side, so it's going to be what we call amphipathic. That means it's going to interact well with both water-loving molecules and water-fearing molecules, right? Water-loving being hydrophilic, water-fearing being hydrophobic. So because it has both regions, it can kind of go across the border and make peace. All right, so bile is really important, again, for the transport of fats through the bloodstream. Other accessory organs of the digestive system are going to include the pancreas. The pancreas is going to have endocrine and exocrine cells. What do I mean by that? Cells that are going to secrete internally and cells that are going to secrete externally. So endocrine cells are going to secrete glucagon and insulin. That includes the alpha and the beta cells. We also have exocrine cells that are going to secrete pancreatic juices. Again, that pancreatic juice goes directly into the digestive tract. It's going to contain bicarbonate ions, which is going to help get our pH back up to 7, as well as enzymes that are going to help continue the process of digestion of the major biomolecules as we travel through the tract. In the small intestines, we're going to enter into 3 regions. The first is the duodenum, the second is the jejunum, and the third is the ileum. The duodenum is what we were talking about when we talked about the secretion of the bile and the pancreatic juices. It's also going to be where most of the absorption of nutrients and water occurs. In the small intestines, we're going to have both mechanical and chemical digestion. And we're also going to have a high surface area of absorption. Why? We have these specialty regions that are called villi, or little finger-like projections. And on the end of those villi, we have microvilli, which are little finger-like projections on the end of the fingers. And these allow us to have a greater surface area, so everything washing across it can be absorbed if it wants to be. The cells of the small intestines are also going to secrete hormones, mucus, and enzymes. And then as we head out of the small intestines, we're going to be leaving from the jejunum. We're going to be leaving from the ileum. So the jejunum is the middle, the ileum is the end. We're going to be leaving from the ileum into the Ileocecal Canal, and the cecum is going to be the beginning of the large intestines. So this is an overview of the small intestines. It has many, many different folds. At the end of these little folds, we're going to have villi. At the end of the little villi, we're going to have microvilli, and that means that we're going to be able to have a really large surface area as the food is going to be passing across, as the chyme is going to be passing across. Okay, as I mentioned previously, we're going to have multiple layers here, including the epithelial layer, which is going to be the lining here. This is going to be simple columnar cells. It's also going to have goblet cells, so we can secrete mucus, etc. And then after we go past this, after the epithelium, is where we're going to find our microvilli, or brush border enzymes are found there at the microvilli. As we enter into this region, we have the villus itself. The villus is going to have a lacteal in the center. The lacteal is going to have both arterial and venous return, so it's going to have a blood supply, as well as this lymphatic tissue, which is where we're going to be absorbing any fats. So anything that's water soluble goes right into the bloodstream and can get absorbed, but all the fats are going to get picked up by this lacteal and go in through the lymphatic system to get dropped in through a different mechanism. All right, continuing down, we have the muscle layer. Here's the muscularis mucosal layer. Here's our circular muscle layer and our longitudinal muscle layer. Again, these are going to be controlled by these different plexus, including this enteric plexus here and the plexus found here in the myenteric region. All right. Okay, so now we're entering into the large intestines. Again, we're doing that through the Ileocecal Sphincter into the Ileocecal Canal. So here's the ileum headed into the cecum. The cecum is going to be the very first part of the large intestines. Heading up, we're going to the ascending colon, the transverse colon, the descending colon, the sigmoid colon, rectum, and the anus. I also want to take a moment to acknowledge this little dangly organ here called the appendix. It's what's called a vestigial organ. We no longer have use for this organ, but it does sometimes become inflamed and does need to be removed. It can cause great terrible pain, and if it ruptures after becoming seriously inflamed, then all of a sudden you can have fecal matter spilling into your abdomen, which can be terminal if not treated. All right, so the moral of the story is, if you feel like you have appendicitis, you should go see a doctor. It should not be ignored. Anyway, food is going to be coming in here through the Ileocecal Sphincter, and as it travels through, it's going to go through what's called haustra. Each one of these are little haustra, right? And the house door is going to have churning within itself, like a little cauldron churning, but also going to push itself forward through each one of these at a time. And so as we move to the next cauldron, we get further and further and further. We're removing the very last of all the nutrients, as much of the water as possible, until we finally end up having what's called a defecation reflex, when a large amount of this fecal matter is going to spill out of the descending colon into the rectum, which is normally empty. Once the rectum is full, we're going to have a distension here on this external and internal anal sphincter. The internal anal sphincter is not going to be under our voluntary control. So it's smooth muscle, and when it has pressure on it, it opens. But the external anal sphincter, that one is under voluntary control. So this one gives you the urge, and this one you can actually release in the bathroom itself. Okay, so again, the ileocecal sphincter is located between the small intestines and the large intestines. The first area of the large intestines is called the cecum. And we in humans have it a lot smaller than animals that eat a ton of grass. In the cecum, we're going to be using this for digestion of a lot of fiber, etc. And so if we have a high fiber diet like grass, then we'll have a large cecum. We don't actually need a large cecum because we as humans are going to be what we call omnivores, which means that we also are going to be eating, although we do eat fiber, we're going to be eating a lot of meat as well. Large intestines are going to include the cecum, ascending, transverse, descending colon, sigmoid colon, rectum, and then anus. Again, there's two sphincters, the internal, involuntary control, external, under voluntary control. We can only control the external sphincters. All right, so what's the purpose of the large intestine? Absorption of water. We've pumped liters and liters of water into our body, and we want to pull them back out before we secrete that fecal matter out. And we also want to store that waste until the time that we are actually able to choose for defecation. And we have specialty bacteria in there, normal flora, that are going to help us with the process of digestion. So let's talk about that gut flora, right? Most of the flora in the colon are going to be different types of bacteria. The dry mass of feces is up to 60% bacteria. So 40% of it is going to be fiber and things that we're unable to digest and water, but 60% of it is going to be bacteria. There's over 1,000 different species of bacteria that are found in the flora of the colon, and depending on which one takes over, you can have different types of intestinal distress. So oftentimes if we have what's called idiopathic means we don't really know why. So if we have idiopathic problems with digestion, like Crohn's disease, for example, so sometimes we can do what's called a fecal transplant from someone who has normal digestion, where we take the normal fecal matter from them with the different species and reintroduce them to someone who may have had their gut flora skewed in one way to another. And that can restore the regular function of the gut flora, including the fermentation of certain undigested substances like the fibers. This incidentally is what's going to lead to our gas excretions in the terms of our farts, right? Methane excretions. We also can involved in regulation of the immune system to toleration of food molecules. So something you may never have seen before, you might have a hard time digesting, but after introduction to it many, many times, your body can start to digest it because the gut flora are able to tolerate things they weren't able to tolerate before just by specifically being introduced to it over and over. Also, by having the right bacteria there, you can prevent the growth of having the harmful bacteria there, right? So making sure that you have the correct gut flora in place means there's nowhere for the harmful bacteria to grow into. And the gut flora help us produce different things like vitamin K, biotin, etc. And they can increase the intestinal cell growth and help maintain that healthy mucosal layer that we need. So again, the gut flora are very, very important. They're also going to be responsible for the creation of lactic acid during fermentation. That's going to help lower the pH, and that's going to help change the growth of pathogenic species. So if we have a lower pH, we're going to help prevent the growth. We've changed the environment so the pathogenic species don't like it. It's not a good host. And they convert carbohydrates into fatty acids that can then be absorbed. So it's going to be the last step of absorption is going to be carbohydrate absorption in the large intestines. Also responsible for taking minerals out of the food and releasing them, things like magnesium, calcium, zinc, and iron that can still be bound up in that residual waste. And they can help metabolize carcinogens. So if we have specialty intestinal bacteria that we know is healthy, that's been linked to the prevention or the reduced incidence of colon, breast, and prostate cancers. All right. So let's talk about what we're digesting. We've been talking about the process of digestion and where all the different organs are, but let's talk about what we're actually digesting. We are digesting macromolecules. Now, macromolecules have both a monomeric form and a dimeric or polymeric form. So monomer meaning 1, di meaning 2. You could also have a trimer, which would be 3, but that would be very specific. Or you could have a polymer, which just means more than one, right? So we have multiple different macromolecules that we can digest, including proteins, fats, nucleic acids, and carbohydrates. We have complex carbs and simple carbs, including disaccharides and monosaccharides. When we break down proteins, we break them down into amino acids. So basically what we're saying is this is the molecules that come in to the body in the burger, and these are the molecules that are able to be absorbed across the intestines to get into the blood supply. So we have to break proteins down into their amino acids. We have to break triglycerides down into glycerol and fatty acids, nucleic acids down into nucleotides, complex carbs into glucose, disaccharides into glucose and fructose, or glucose and galactose, or whatever they're comprised of, right? So we want to get down into the monomeric form of this sugar, which includes glucose as a monomer, fructose as a monomer, galactose as a monomer, etc. So in order to do that, we have to start, we're taking something very large and we're breaking it down into something very small. So we have to start in the mouth, and then we increase this in the stomach, and we are going to continue this in the intestines, etc. So it's going to be something where we're breaking things down in many, many stages. So we're going to be breaking large things down into medium size down into small sizes. So we have the very first things that we're going to hit in the saliva is going to be amylase and lipase. Amylase breaks down amylose, which is a sugar or a starch. So we're going to break down a starch into a sugar. Saliva is going to be a lipase. Salivary lipase, is going to break down fats. So lipase breaks down lipids, breaks down fats. In the stomach, we're going to start breaking down proteins for the first time. So in the stomach, we're going to see gastric pepsin, which is going to help break down proteins. And the pancreas is going to secrete a ton of enzymes as well, including amylase, which is the same that we saw in saliva, but instead of it being salivary amylase, now it's pancreatic amylase. That breaks down starches. Pancreas also is going to secrete lipase, which breaks down lipids or fats, ribonucleases, which breaks down RNA, deoxyribonucleases break down DNA, as well as trypsin and chymotrypsin, which start breaking down proteins. We also have carboxypeptidase, which breaks down peptides. Proteins are the large version, peptides are the smaller version. We want to get down to amino acids. So this breaks down large into medium. This breaks down medium into small, if that makes sense, right? Okay, in the small intestines, we're going to be breaking down medium into small. We have sucrase being, it's an enzyme called sucrase, which breaks down sucrose. This is a dimer. We just saw here, it's going to be a dimer of glucose and fructose. We're going to be breaking it down into monomers. Again, the monomers are what are going to be able to be absorbed across the digestive tract in the intestines. So again, these are examples of different enzymes that are going to break down dimers of sugars into the monomers of sugars. And then peptidases are going to break down into peptides, or break down peptides into amino acids. So again, in the small intestines, we are seeing medium into small, whereas in specifically in the mouth, we're seeing large into medium, and in the stomach, we're also seeing large into medium. In the small intestines, we're seeing medium into small. All right, so let's talk about the process of absorption. Again, we can only absorb the smallest of the micronutrients. The small intestines is going to be arranged intentionally, anatomically intentionally, so that we can have a larger surface area because we have the villi and the microvilli. So the villus is going to increase surface area, and then on top of the villus is the microvillus, which increases surface area even further, and form brush borders, which have specialty enzymes called brush border enzymes, which break things down as they pass by, allowing them to be absorbed. So here's the villus. At the end, we have the microvillus. At the tips of the microvillus, we have brush borders. So if you picture this as a finger, these are fingers on fingers and fingers on fingers on fingers, right? So the microvilli are going to be little teeny tiny regions that are going to come off of the villi, and the villi are going to be coming off of the edge of this lacteal region. So inside here we have, again, the lacteal region, which is going to have the lymphatic tissue, absorption of fats, as well as vascular tissue, which is going to allow for the absorption of water-soluble nutrients. All right, so GI tract is going to have a lot of immune system and nervous system component. Why? Well, what does the immune system do? Well, we just ate something from the outside. It might not have been cooked properly. It might have been contaminated. It might have had bacteria on it. We want to make sure that we kill anything that's a foreign invader. And it also has a really large nervous system component because we want to make sure that we're contracting in time, right? We want to make sure that we're pushing things across and that everything is given the right amount of time in each of the stages of the organs, right? Of each of the regions of the digestive tract. Now, serotonin is going to be produced by most of the neurons in the digestive tract. Almost all of the serotonin in the body, 95% of the serotonin in the body is produced by the digestive tracts. And we also have lymphatic tissue within the digestive tract that are known as lymph nodes, as well as, we learned it in growing up as mucosal-associated lymphatic tissue, MALT, but now it's known as GALT, or gut-associated lymphatic tissue. It's going to be lymphatic tissue that is going to be found throughout the mucosal layer of the digestive tract in little nodules, basically. Right, so here we're looking at the absorption of nutrients. Here's fructose. It can be absorbed directly. Glucose or galactose can be absorbed directly, right? And then once they get sent in, they just go directly across and facilitated transport into the blood vessels. So sugars have no problem going across. These are the microvilli. This is the columnar epithelial cells of the intestines. Sugars have no problem going right on in to the cell, across the cell, and being sent right back out, and then picked up by the blood supply. The same thing can happen with small peptides and amino acids, although in this case we're going to be negotiating moving the sodium in. Here we're going to be moving sodium in and hydrogen out. And we also are going to be using ATP to be bringing in potassium and pumping out sodium. So the sodium-potassium pump is going to be using ATP. So it is going to cost us a little bit of energy for absorption to occur, but my point is that we're able to bring it right into these blood vessels, whereas something like fats is not going to be able to do that. So something that's a fat-associated molecule is going to have to be picked up specifically and sent out through the lacteal region, through the lymphatic system. All right, so nucleotides, which is the smallest version of nucleic acids, are able to be picked up as well. Again, we still need that sodium-potassium pump running. Why? Because we're using sodium to import the nucleotides. So we end up with too much sodium in the cell. Got to pump the sodium out. Got to bring the potassium back in to do that. So we keep it as a positive charge, or keep it as a neutral charge. And then the nucleotides, again, they're going to go straight across and into the blood supply. Same thing with ions and water. They should be able to go right on in and be picked up by either paracellular or transcellular or facilitated transport to go into the blood supply. However, fats, on the other hand, now we're talking a different story. Fats are going to be broken down by bile. Again, this is made in the liver and stored in the gallbladder. And they're also going to be broken down by phospholipids, which are found in food, into smaller droplets. This process of this is called emulsification. So we make it smaller and smaller and smaller fat droplets until they're able to be chemically digested. So they're physically digested and chemically digested as well. So they can be chemically digested by the pancreatic lipase, which is an enzyme which breaks down lipids. Anytime you see ACE, you know enzyme. In this case, ACE following lip means that it's an enzyme that breaks down lipids. Pancreas is also going to secrete something called colipase that binds to the lipase and to the lipid droplet, and that makes it a smaller and smaller and smaller droplet until we finally make something called a micelle, which is very, very tiny. Now, a micelle is going to be able to transport these fats across. This is going to be here, a micelle here. Going to help us transport fatty acids directly across the intestines of the epithelial cell of the epithelial cell of the intestinal lining. However, it's still not going to be able to go directly into the blood supply. Why not? Because the blood supply is water soluble and these guys are fat soluble. Okay, so in order to absorb that fat, again, we're going to break it down into a droplet or a micelle, absorb it across the epithelial cell, and then form triglycerides in the smooth ER, then get transported through the Golgi, through LDLs, again, which are going to be made by the liver, so through a vesicle that's coated by LDLs, and then it's going to exit out into the lacteal. And again, in the lacteal region, we're going to be picked up by the lymphatic tissue, transported and dropped into the bloodstream through the thoracic duct, where it's able to translate or to circulate through because of the LDLs, which are, again, amphipathic molecules, or molecules that have both a water-soluble and a fat-soluble region. Okay, so this is what I'm talking about when I'm talking about the absorption of monoglycerides, cholesterol, and fatty acids. Anything that is fat-soluble can be broken down, diffused into the cell, but needs to then be processed by the ER and the Golgi and secreted out in a chylomicron. What does that mean? This chylomicron here is going to be able to be carried out through the lymphatic tissue and then dropped back into the blood supply through the thoracic duct, where it can be carried because of LDL molecules, which will help carry it through the aqueous solution. So that includes fat-soluble vitamins like A, D, E, and K. But water-soluble vitamins are going to be absorbed just in the regular way by diffusion or by mediated transport, with the exception of vitamin B12. Vitamin B12 is kind of special. Remember, it has to be bound to a transport protein that's known as intrinsic factor. The intrinsic factor is going to be secreted by the stomach, right, the parietal cells of stomach, and then absorbed in the ileum. And when we bring that in, that B12 deficiency, when we bring that B12 in, we're able to absorb it only if we have intrinsic factor. So if we have enough B12, but it appears as though we are deficient, we could be because we don't have enough intrinsic factor. And if that's the case, we need to actually be giving yourself extra B12 because your body's not able to absorb all of what it has. So it's only absorbing a small percentage of it. And again, if we have B12 deficiency, it can turn into pernicious anemia, which means that you have a very low iron count, which can lead to low red blood cells. Okay, so let's talk about nutrient transport as an overall, right? So this is the villus. It's extending into the lumen of the intestines. We're going to have on the end of the microvilli with little brush border enzymes, and we're going to have absorption of nutrients across these epithelial cells into this lacteal region. Inside here, again, we have the lacteal. The lacteal is going to be a lymphatic tissue, so we're going to be picking up any fats. You can see those shown in yellow, little yellow circles. And any water-soluble molecules will be brought in, like amino acids and sugars. Those are all going to be brought in, monosaccharides here through the venous return. So again, here's the arteries and the veins, going to be brought in through the vasculature. The vasculature drops directly into the hepatic portal vein, right? So all food and nutritions go through the liver. Why? We hope we didn't miss any bacteria, but if we did, or any toxins, but if we did, the liver is going to take care of it for us before this is going to get dropped back into the bloodstream, right? So inside the hepatic portal vein, we're going to go through, do some processing in the liver, and then drop the nutrients back into the blood supply. Here, we've got the fat-soluble tissue, molecules coming in through the lymphatic vessels, and they're going to get dropped into the thoracic duct directly into the vasculature. Okay, so how do we control digestion? And we do that by controlling the amounts that's inside the lumen, the volume and composition of the luminal contents. So that means the body is designed to absorb all nutrients that are ingested, whether we need them to function or not. Whatever passes through, it absorbs. And we have three major phases of digestion. Cephalic means brain, gastric means stomach, and intestines mean, well, intestinal, right? So cephalic phase is when we are going to have the stimulation of digestion by the thought of food, the smell of food, the sight of food, or the actual taste and chewing of food. So this can be the example of like when you really aren't hungry all day long until you get home and you smell the food that your roommate is making and you go, oh, I actually am hungry. And all of a sudden, that's the cephalic phase. That's when your head says, I'm ready to eat. The gastric phase is going to be initiated when the food comes in contact with the stomach lining. So after swallowing, down the esophagus, through the Gastroesophageal Sphincter, also known as the lower esophageal sphincter, into the stomach, we're going to initiate the gastric phase. And we do that by increasing the acidity of the parietal cells. We're going to put a bunch of HCl in there. And we're going to have the presence of amino acids or peptides in the stomach. So any of those can initiate the gastric phase. And the intestinal phase is initiated by the stomach pouring its contents into the small intestines. So as soon as we get stimulation in the small intestines by either distension or an acidity, which would happen if it came out of the stomach, right, or an osmolarity and or the nutrient contents in the intestines. All right, so this is what gastric glands look like. These gastric glands here are going to have pits. At the bottom of them, we're going to have regions that are called the enteroendocrine cells that produce gastrin. Coming up the sides, we have chief cells that produce pepsinogen, and we have the parietal cells. Remember, the parietal cells are going to produce intrinsic factor and hydrochloric acid. They don't produce the hydrochloric inside the cell. Rather, they're going to secrete right next to each other hydrogen and chlorine, and because of the fire hose of both of them, it's going to turn into hydrochloric acid inside the lumen. We also have goblet cells up here that are going to produce mucus, and all of that is going to get secreted out that gastric gland into the gastric lumen. All right, so we are also going to have neural regulation of digestion, and we do that through the enteric and the central nervous system. So the enteric nervous system is going to innervate the submucosal plexus and the myenteric plexus that I pointed out earlier. Remember, the submucosal plexus is going to be involved in the secretory pathway, and the myenteric plexus is going to induce the contraction of the smooth muscle. So it's going to coordinate the muscle contractions of the circular and the longitudinal muscles. And the central nervous system is going to contribute to the neuronal control of the gastrointestinal system through regulation of either sympathetic or parasympathetic systems. Remember, sympathetic is going to be fight or flight, and parasympathetic is going to be rest and digest. So under the parasympathetic control, the gastrointestinal system is going to be upregulated. But if you get a really good scare that sends a bunch of adrenaline through your body, the gastrointestinal system can be downregulated so that you can focus on fight or flight. And a hormonal regulation can also be involved in the regulation of digestion. So This is just for you guys to make a chart at home, but I can talk you through it right now. So where are these different things produced and what are their functions, right? So I'll just start with the first one, right? Gastrin is going to be found in the stomach, and the function is going to be involved in feelings of satiety, right? So regulation of digestion is involved. It has multiple different sets of chemical stimuli that are going to help stimulate your brain into thinking that you're full, or telling your body that you need to start stimulating things that are going to help digest, etc. So leptin is something that's secreted by fat cells, and it's going to decrease the hunger center in the hypothalamus. So what does that mean? If you have a ton of fat cells in your digestive tract, it's going to say, I'm not really hungry right now. I'm still working on the fat, right? So we also have ghrelin, which is secreted by the stomach lining, and also is going to be increased during fasting, which is going to stimulate the hunger center in the hypothalamus. So again, the hypothalamus is a region of the brain that is going to control whether or not you feel hungry. These are all going to be different disorders of the digestive tract, including the reflux disorder. So this is going to be esophageal reflux disorder, when we're going to have heartburn because of the lower esophageal sphincter might be leaking. So we might end up with some of that gastric juices up into the throat. We can also have a hiatal hernia, which can happen when we have a hernia inside the intestinal lining. We can have ulcers. Ulcers occur when the bacteria kind of make a little pit and start digesting the skin cells that are lining the epithelial lining of the stomach. And when that happens, it can also happen in the intestines, but it's mainly in the stomach. And when that happens, if we have ulcers, then we can end up with the hydrochloric acid layering on top of the ulcers, which makes it even worse. So we have a cycle where we end up with damage that doesn't get repaired and then gets further damaged. Crohn's disease is going to be something that's going to be what we call idiopathic. There's something wrong with your digestive tract, and we're not exactly sure what. We do know that oftentimes in these cases, a fecal transplant has been super helpful. So perhaps their fecal bacteria levels aren't exactly correct. The fecal flora, we have something bad in there that's overtaking, or not enough of the right thing. And so if we put in something from someone who's healthy, generally we can restore their, or not always, but often we can restore their digestive function. Ulcerative colitis is going to be when we have ulcers in the colon. So again, this is going to be pockets of epithelial tissue that are damaged and destroyed because of bacteria usually, which in this case are then going to get inflamed even further because of fecal matter in the tract. Gastritis, so infection of the gastric tract. Diverticulitis, when we have a region, generally this is going to stem off of ulcerative colitis. So if we have ulcerative colitis, we can have a region where the fecal matter is supposed to be traveling across it, but instead we have a pocket that pools and becomes a larger ulcer region, and eventually becomes larger and larger until we end up diverting that fluid out into a secondary pathway, which can turn into a leakage, etc. So it can be very tricky. Irritable bowel syndrome is going to be placed in that idiopathic region with Crohn's disease. Again, can also be treated with a fecal transplant. And hemorrhoids, well, normal hemorrhoids are going to be internal. So everyone has hemorrhoids. Hemorrhoids are not something that are an abnormal feature, but having them on the outside is abnormal. And if, in this case, there's ways that we can shrink them or put a band-aid around them to help them fall off, or learn how to reinsert them, but they can be incredibly painful. All right, so these are some disorders of the digestive tract. Here is going to be a hiatal hernia. You can see we have a piece of the intestines actually popping in through another region, causing a blockage. Here we have an ulcer. So the ulcer is going to be a region of the epithelial lining that has been degraded. And therefore we have pockets where we can have either fecal matter or intestinal juice or stomach acid aggravating that. So it further gets, makes further issues. And then diverticulitis, this is what I was saying. We have little pockets where things that end up pooling in there that can eventually turn into separate alleyways that run to nowhere, and things can end up stuck in here, causing major problems. So these are major disorders of the digestive tracts. Okay, so some of the effects of aging. So as we get older, our digestive tract stops working as well as it did when we were younger. And as we get older, constipation is something that can be an issue for elderly people, especially if not enough fluid is consumed. And we can end up with problems with nutrient absorption. So we need to have a higher nutrient content in a lower calorie diet. And they can have problems with intestinal lining being damaged. We just show you some images of that. Polyps can occur, and that esophageal reflux disorder is also more common in the elderly. Okay, so last but not least, let's go back to the application question that we talked about when she was digesting the food. So the mechanical digestion is going to occur in multiple regions of the digestive tract, including the mouth, stomach, and small intestine. Chemical digestion occurs in the mouth, stomach, and small intestine as well. Which one of these different types of food are going to have the different amounts of biomolecule? Obviously, the beef is going to have the highest amount of protein. The highest amount of fat is also going to be in the beef, but some in the broth, starch in the potatoes and carrots, fiber in all of the vegetables. Which enzymes break down what? Well, protein is going to be broken down by pepsin, trypsin, and chymotrypsin. We're also going to be able to, that's going to be in the stomach. And we're also going to break down peptides like carboxypeptidase, and peptidase is in the small intestines. Amylase breaks down starches. Maltase breaks down maltose. Anytime you see ACE, it's going to line up pretty well with its OS, very likely. So maltase is an enzyme that breaks down maltose. It's going to be the sugar. What breaks down fat? Lipase, right? Lipase breaks down lipids. And the feeling that's going to make Martha feel satiated is by cholecystokinin, or CCK. So thank you so very much for listening to our lecture today, and I can't wait to see you guys in class again next time. As always, happy studying and have a great day.